The method combines EEG and TMS for real-time brain state-dependent brain stimulation. This enables the TMS pulses to be synchronized with a specific phase of ongoing endogenous brain oscillations.
Brain stimulation is a technique for modulating the brain. By synchronizing individual TMS pulses with a specific EEG-defined brain state, the effect of the stimulation can be optimized.
Standard TMS is already used to treat neurological and psychiatric diseases such as stroke and depression. EEG-synchronized TMS has the potential to improve treatment outcomes using personalized stimulation protocols.
We also use EEG-synchronized TMS to investigate the basic neurophysiology of brain oscillations in the human by comparing the effects that stimuli have, which are identical but are applied during different brain states. The effects can be subtle and are easily washed out by other sources of experimental variability. We found that low stimulus intensity works best to observe the effect of sensory motor rhythm on corticospinal excitability.
To conduct a closed-loop experiment, brain activity is recorded through EEG potentials from the scalp surface that are digitized by an EEG amplifier. The signal then passes to the EEG main unit, and from there to the real-time device which controls the stimulator, thereby closing the signal loop between measuring and modulating brain activity. The real-time device analyzes the EEG oscillations and sends a trigger signal to the TMS stimulator when a predetermined trigger condition is